topic is on immunoendocrine interactions, implications for health and disease. And this is a branch of biology where the two disciplines have been merged together. Like the immunology is one separate branch and endocrinology is another branch. These two branches are merged together, and this hybrid discipline was mainly pioneered by a scientist, an Italian scientist called as Calzolari. So he is regarded as the father of immunoendocrinology because of his work related to the immunoendocrinology started with the castration of the rabbits. Following the castration, that is the removal of the gonads, there was enlargement in the thymus gland. And this thymus gland is acting as both endocrine gland as well as the part of the immune system. And this gland is located within the heart, the chest cavity, just in front of the heart. And this gland produces certain hormones like the thymopoietin, thymosin, etc. So these hormones are also involved in the regulation of the release of the T lymphocytes, because thymus is the source where the T lymphocytes will undergo maturation, differentiation as well as the maturation. So with this, there were several other works which followed, which resulted in several discoveries, particularly the involvement of several endocrine glands in regulation of immune system came into the picture, like the hypothalamus, pituitary gland, gonads, adrenal, pineal, thyroid, and thymus. All these glands are known to be involved with the immune regulation. So I will just quickly go through the organization and function of the immune system. And we know that there are two kinds of lymphoid organs. One is a primary lymphoid organ, the other one is the secondary lymphoid organs. Primary lymphoid organs include the thymus gland as well as the red bone marrow, where these are the sources of the lymphocytes, like the T lymphocytes are being produced from this red bone marrow. The B lymphocytes undergo maturation within the red bone marrow only, whereas the T lymphocytes migrate to the thymus where they undergo differentiation and maturation. So on the other hand, the secondary lymphoid organs are there. These include the lymph nodes, which are located in various parts of the body or regions of the body, like the neck region, armpit, as well as the groin region. And in addition to that, the spleen also acts as a secondary lymphoid organ, and the Peyer's patches within the intestine. And even this appendix is also considered as one of the secondary lymphoid organ. And in general, these are all considered as the gut, that is gut-associated lymphoid tissues. Now, there are two kinds of immune responses. One is innate immune response, which is also known as the non-specific immune response. Means this is works on a broad spectrum. Like this is not specific against a particular pathogen, but it works on all sorts of infections. So this is considered as the first line of defense, where our body is protected by the epithelial lining, both the external surface, that is through the skin, and also internal linings, like the lungs lining is also provided with the epithelial lining, and also the gut is also provided with epithelial lining, which do secrete mucus. And these are mainly responsible for fighting with the pathogens. So if the pathogen has to invade our body, it has to pass through that first line of defense. And in case if it is successful in passing through the first line of defense, then it has to face the second line of defense, that is called specific immune response. And these specific immune responses are specific against a particular pathogen. And this type of immune response is also called as acquired or adaptive immune response. So these are of two kinds: cell-mediated immune response and antibody-mediated immune response. Cell-mediated immune response mainly involves the T cells, whereas the antibody-mediated immune response involves the antibodies produced by the B cells. So B cells are involved here. This is also called as humoral immunity. And for our convenience, we differentiate these cell-mediated and antibody-mediated immune response. But if you consider the total immune response, these two are often interconnected. So here is the brief overview. the antigen is the source which is capable of causing the infection. So this can be coming from the microorganisms or any other allergy causing substances. So once these enter inside our body, the macrophages will recognize this, and they will interact with these antigens, and they destroy, they degrade, and they display the antigenic substances on their surface. So these are called as antigen presenting cells, and these cells are now recognized by the cytotoxic T cells, and they will start attacking on those pathogens which are displaying such kind of antigens. And the helper T cells, these are the most important cells which are known to further accelerate the immune response. So these helper T cells recognize those macrophages, and they stimulate the cytotoxic T cells as well as they stimulate the B cells. So this is where both humoral immune response as well as the cell-mediated immune response both are interconnected. So the B cells will produce the antibody, and this antibody antigen complex is formed so to nullify the action of the antigens. So this will also result in the subsiding of the effect of the infection by the antigens. So here there are also other type of cells called as the suppressor T cells. So these suppressor T cells are also known as the T regulatory cells. So these T regulatory cells are known for suppressing the immune response. They will calm down the immune response so that further immune response will not be continued. Otherwise, the immune system can be overactive, and that can keep on destroying our own cells also. That is prevented by through these T regulatory cells. So there are also another type of cells called as killer cells or natural killer cells, and these killer cells are also known to be responsible for production of certain cytotoxic chemicals, and thereby they can kill the pathogens. Okay, so here the thymus gland is a bilobed structure, and here is the sectional view of the thymus gland, where you will see it is covered by the capsule, the outermost connective tissue capsule, and inside there are several lobes, and these lobes are separated by means of trabeculae, and each lobe has got two parts here. One is cortex, the outermost cortex, and innermost medulla region. And here is the histological view of the thymus gland can be seen, and this darkly stained portion is known as the cortex, and the lightly stained portion is called as medulla. And this is the enlarged view, where the thymus cortex is known to contain mostly the immature T cells, whereas the medulla portion mainly contains the mature T cells. And in the thymic cortex region, there are also some cells which are shown with the arrow mark here. So these are called as the reticuloepithelial cells. So these are the cells which are concerned with production of the hormones, endocrine hormones like thymosin, beta 4, then thymolin, thymopoietin. And in the thymic medulla, these large structures are also known as the Hazel's corpuscles. And these Hazel's corpuscles are also known to be involved in the maturation of the T lymphocytes. Now, there is a syndrome called as a Dye-George syndrome, which is a congenital disorder. That means this disorder occurs at the time of the birth itself. And this particular disorder is characterized by the absence of the thymus, or the thymus is underdeveloped. So in this kind of condition, the newborn infants will suffer from a severe immunodeficiency because of the absence of the thymus gland. Because the thymus gland is the gland which degenerates with the age, but at the time of the birth, the thymus gland is quite larger. So it weighs about 70 grams at the time of the birth, whereas after 60 years, it is reduced, or in general, in adult stage, it reduces up to 3 grams. So there is a degeneration of the thymus gland. But by the time of the birth of the individual itself, the thymus gland has already produced all the required T cells that are needed for the sufficient immune response. So if the baby is born without the thymus gland, then it has to suffer from severe immunodeficiency. So in such cases, if there is any transplantation done from stillborn baby. Stillborn baby means the baby has died at the time of the parturition itself, but still the baby's thymus gland is developed. So in such cases, if the thymus from such babies are transplanted to the baby suffering from the Digeorge syndrome, that can lead to a condition called as graft versus host disease, where it will start attacking on the cells of the recipient. So this is otherwise known as the graft versus host disease. Okay, now whether the male as well as the female exhibit any sexual dimorphic immune response. Who has more immunity, whether it is a male or female, is the question here. So there is a sexual dimorphic immune response because of the variations in the hormones between the male as well as the female. Obviously, there are some differences that can be noticed between the male and female. Especially the female is having the highest immune response as compared to the males. So cellular and humoral responses in non-pregnant healthy adult females are generally more active than those in healthy adult males. Many in vivo and in vitro studies in both humans and animals have found higher levels of IgG, IgM, and IgA. These are all the class of immunoglobulins which are found in higher concentration in case of the females than in case of the males. And here is the table which clearly illustrates the differences between the male as well as the female with reference to the type of the immune cells. The size of the thymus is larger in case of the males, and also the CD8 plus T cell counts are higher in case of the males. CD8 plus means these are the T cytotoxic cells. So their counts are high, and the size of thymus is also larger in case of the males. But in case of female, the CD4 plus T cell counts is high. So CD4 plus means these are the T helper cells. Their number is high in females, and even the CD4 CD8 T cell ratio is also higher in case of the females, and the number of activated T cells are also higher in case of females. T cell proliferation capacity is also greater in females, and antigen presenting efficiency is also higher in case of the females. So because of all these reasons, in general, the females are capable of exhibiting highest immune response compared to the males. If there are any challenge with antigens like the poliovirus, it can stimulate the higher titers of antibodies in females than in case of males. And even the skin graft rejection time is more rapid for females than for males. Skin graft rejection means, suppose if a portion of the skin from the donor is transplanted to the recipient, and the recipient, if it is not compatible with such kind of transplantations, they reject altogether the grafted tissue. So this kind of rejection time is more rapid in case of the females as compared to the males, which clearly tells us that the female's immune response is highly active. However, this sexual dimorphism is frequently not seen until puberty. And you know the reason very well, that until the puberty, the hormones concerned with the reproduction are not well developed. So that at the time of the puberty, the hormones responsible for reproduction are highly active. So then the sexual dimorphic nature becomes more prominent after the puberty. Okay, so the immunocompetence of cycling female and the window of vulnerability here, there are possibility of the fluctuations in terms of the sex steroid hormones in case of the female during the menstrual cycle, because during the phase of the menstrual cycle, the estradiol hormone concentration is not the same. So there is a fluctuation that involves the production of the estradiol hormone as well as the progesterone and the FSH hormone as well as the LH hormone, which are all actually being regulated, that will be responsible for triggering the ovulation and also following that the maintenance of the pregnancy. So because of these fluctuations during the menstrual cycle, the immune response also is subjected to variation. If you can see in this picture, the The menstrual cycle is characterized by three major phases, like the menstrual phase, which is characterized by the discharge of the menses, which can occur up from 5 to 7 days, and this is followed by the proliferative phase, and that can occur up to the 14 days, and then that is followed by the secretory phase. So here, the proliferative phase is characterized by the rise of the estradiol level, and this increasing estradiol level stimulates the LH, and so that the LH surge takes place. Following this LH surge, the ovulation occurs. So this is characterized by the decreased estradiol level and highest level of LH. So once the ovulation takes place, then the estradiol level is decreased briefly during the secretory phase, and then that is followed by a brief rise. Whereas the progesterone hormone reaches the peak during the secretory phase. So this particular variation with reference to the estradiol as well as progesterone is also playing a regulatory role on the immune response in case of the female, that is within the different phases of the menstrual cycle. So if you can see here, the proliferative phase particularly is characterized by the decreased infectious susceptibility because of highly active cytotoxic T cells here, and also the presence of immunoglobulins like IgG and IgA are also higher, and the T regulatory cells will also be higher by the end of the ovulation. But following the ovulation, there is a decrease in the IgG and IgA, and also this endometrial cytotoxic cells activity, and also the CD4, that is a T helper cells activity. This is in response to the increased level of the progesterone as well as the estradiol here, so that the infectious susceptibility will be higher during the secretory phase. However, in the lower reproductive tract, the T cytotoxic cells activity remains still higher in order to prevent the pathogenic effect, any entries. But in general, during the secretory phase, there is a decreased immune response. This is because of the increased progesterone as well as the estradiol hormone level. But this is like a cycle. This will be continuous. That will be happening. But if the pregnancy occurs, then there are a number of other changes that can be observed here, because the pregnancy is a quite complicated event, which involves the development of a baby inside the mother's womb, and this baby is considered as the semi allograft. Like it is also carrying the antigens coming from the paternal source, that is from the father's source, because of which the mother's body can treat the baby as the foreign one. So there is a possibility that the developing baby may be rejected by the mother. So our immune cells should be calmed down. So our immune cells should treat this baby as the own, and you should not differentiate this as the foreign baby. So this kind of regulatory mechanisms are very much required in order to achieve a successful pregnancy. So immune response during pregnancy is complex and depends on the stages of the pregnancy. Depending on the stages of the pregnancy, the immune response is also being regulated. So during the first trimester, the natural killer cells, then dendritic cells. The dendritic cells are also known as the antigen presenting cells, and the macrophages, the phagocytes, infiltrate the decidia and accumulate around the invading trophoblast cells. So here, this is a location where the placenta will be taking place. That is in the endometrial layer. The trophoblast cells, which serves as the fetal maternal interface, will invade the uterine epithelium, where it has to establish the suitable environment for the supply of the nutrition from the mother to the baby. So here, the immune response, we cannot say that it is suppressed, but it is still functional during the pregnancy, and it is carefully controlled, although progesterone levels are high here. Okay, so here the paternal antigens, like these antigens are generally known as the HLA-G, human leukocyte antigen, which is coming from the paternal source, are being recognized by the dendritic cells. So these dendritic cells will recognize these antigens from the fetal cells itself, through the trophoblast cells. So they do display these antigenic substances to the T regulatory cells. These T regulatory cells will now, in the presence of interleukins, like these are the cytokines, interleukin 10 as well as the TGF beta. So they will signal the deciduous natural killer cells. So these natural killer cells are generally cytotoxic in nature. And these natural killer cells are present in the endometrium as well. And these endometrial cytotoxic cells, or the natural killer cells, are quite stronger. That means they are producing highly cytotoxic substances. But these decidual natural killer cells are the one which have received the message from these T regulatory cells through these interleukin 10 as well as the TGF beta, so that these decidual natural killer cells are different now from the natural killer cells which are found in the just endometrium. So their cytotoxic nature is decreased, and this actually favors the development of the uh, baby in the mother's womb. Means they are mainly responsible for inducing this immune tolerance. Immune tolerance means the immune system is not responsive to the antigens. So that is called as immune tolerance. So the immune tolerance is a must event that should occur here in order to facilitate the development of the womb. So this event is mainly because of the deciduous natural killer cells, which will reduce their cytotoxic nature in response to the paternal antigens here. So the decidual natural killer cells are cytotoxic in the absence of HLA-G. Suppose if these decidual natural killer cells are not receiving this HLA-G signal, then these are highly cytotoxic. So the HLA-G signal is very important, which is coming from the paternal source, that will signal the T regulatory cells, and then these D natural killer cells. Then the cytotoxic nature comes down. Suppose if this HLA-G is not present, then these D natural killer cells will behave similar to that of the natural killer cells in other parts of the body, and they are highly toxic, and they can even attack the baby in case of the pregnancy. So this is again another picture where the involvement of different cells are being shown here. Like the extravillous trophoblasts are the one which are invading the uterine epithelium, particularly the endometrial layer, and they establish the contact here with the blood vascular system. And these blood vessels are also undergoing some kind of remodeling here. So these are called as a spiral arteries here. And these spiral arteries are mainly responsible for collecting the nutrition from the mother side and transferring these nutrients into the baby through this maternal spiral network. So then there are decidual natural killer cells, as I told before, and there are also the decidual macrophages and dendritic cells, and there are also the decidual stromal cells, then the mast cells, uterine mast cells, T cells, as well as B cells. All these cells are present in the region where the placenta is being established. And here there are mainly three phases, like the pro-inflammatory phase, which occurs during the first trimester, where the initial immune response needs to be calmed down. So that is being established here. And also the formation of this maternal spiral artery, which is mainly through these decidual natural killer cells, which do produce this angiopoietin 1 protein, which is mainly responsible for the formation of the maternal spiral arteries. And the dendritic cells are responsible for the antigen presentation. And these uterine mast cells are mainly responsible for the embryo implantation. And these decidual stromal cells limit the influx of cytotoxic T cells into the endometrium, and they do secrete interleukin 15. And this interleukin 15 is also a very important uh, signal. So the anti-inflammatory stage during the second trimester. So the first trimester is up to the 3 months, and the second trimester there is increased immune tolerance because already the stage has been set during the first trimester itself, and then this is followed by again a pro-inflammatory response during the parturition. So once the baby development is completed and it is ready for the parturition, then this time again the production of cytokines like the interleukin 1 beta, interleukin 6, and TNF alpha, tumor necrosis factor alpha, and interleukin 8 increases during the labor. So these change in the production of the interleukins will trigger the pro-inflammatory responses. So till the time actually the immunotolerance is going on, so that so the immune response is limited. But once this pro-inflammatory response is triggered at the time of the parturition, that will result in the violent contractions of the uterine myometrium that results in the expulsion of the baby. So this is also a very important requirement in order to ensure the normal delivery of the baby. So the immune system helps in successful implantation of the fetus. In the absence of decidual natural killer cells, trophoblast cells are not able to reach the endometrial vascularity, leading to termination of pregnancy. So we cannot say that pregnancy can be successfully completed in the absence of the immune cells, because if these decidual natural killer cells are not present, then the trophoblasts are not able to successfully establish the connection with the endometrium of the uterus. So that can result in the termination of the pregnancy also. Then the depletion of the dendritic cells also prevents the blastocyst implantation and decidual formation. So the immune system also establishing a cooperative role here in order to complete the successful implantation of the fetus and also the pregnancy and the safe parturition. Again, some interesting features are observed here in the first trimester, during which the immune response is quite strong. There can be spontaneous abortions in the first trimester. As compared to the woman who has successfully completed the term. Once this first trimester is successfully passed, the chances of abortion is less. But the chances of spontaneous abortions in the first trimester are quite high. And this is also viewed as one of the important adaptation as far as the evolutionary perspective is concerned as well. So as you can see in this graph, in the first trimester, so this can span up to the 12 weeks period. So during this particular period, the pregnant female usually will develop a kind of nausea as well as a vomiting sensation. And this is also known as the morning sickness. So this kind of morning sickness is mainly because of the changing hormones that are occurring here, that is characterized by the high amount of estradiol hormone as well as the progesterone hormone, which is being secreted by the corpus luteum. And also there are a lot of changes that are taking place within the immune system also, so that they are getting adjusted with the fetus which is being implanted. So at this particular juncture, the female develops nausea as well as vomiting sensation. And this is quite intensive in the first trimester. So it is viewed that this kind of morning sickness is quite evolutionarily important, and it is conserved because during this particular time period, there is development of vital organs in the baby, I mean the embryo. So that includes the CNS as well as the heart, upper limbs, eyes, lower limbs, teeth, palate, then external genitalia and ears. So all these are the most important vital organs, and these are mainly being differentiated during this particular period, that is in the first trimester period. And this morning sickness avoids the intake of any such kind of unwanted food materials that can act as allergen and that can trigger the immune response. And these unwanted substances may interact with the developing baby, so that some kind of anomalies can also be produced, because any contaminants, if they can invade the body, that can reach the developing fetus during this time, and that can result in abnormalities in the embryonic development. So the body is designed in such a way that during this particular time, usually because of the nausea and vomiting sensation, the female avoids intake of such kind of allergy causing food. So that is also one of the adaptation that favors the successful completion of the pregnancy. In case of humans, during the second and third trimesters onwards, decreased number of cytotoxic T cells and helper T cells are observed, and this is a characteristic of the immune tolerance. Pregnant women do not reject foreign skin grafts as effectively as the non-pregnant woman. So in case of non-pregnant woman, the immune response is highly active, whereas in pregnant woman, the immune response is subsided because of the immune tolerance, which is there because of the developing embryo. The clinical studies reveal increased occurrence of smallpox, polio, viral hepatitis, varicella zoster virus. This causes herpes, influenza, then the rubella. Rubella causes measles. Cytomegalovirus, this also causes herpes, and this pulmonary and systemic mycosis. Pulmonary mycosis means the fungus which is causing infection to the lungs, and the systemic mycosis in other parts of the body, mainly the skin portion. So these kind of infections can be increased during the pregnancy because the female is exhibiting immunotolerance during this particular period. 3 to 6 months after the birth of a child, pre-existing maternal autoimmune diseases such as Hashimoto's, thyroiditis, and Graves' disease may be exacerbated. If the female is suffering from the Hashimoto's, thyroiditis, and Graves' disease, and these are the autoimmune diseases, and these are tied with the scientists' names here. Hashimoto is also a name of the scientist, and Graves is also a name of the scientist. So the Hashimoto's thyroiditis is characterized by the destruction of the thyroid gland, where our own immune cells, including the T cells as well as the antibodies, they identify the thyroid follicles as the foreign source, and they will start destroying. As a result, the thyroid cells will come down, and this is a characteristic of hypothyroidism. And the Graves' disease is opposite one here. This is characterized by hyperthyroidism, where this disease is characterized by the hyperactivation of the thyroid follicle cells. And this hyperactivation is mainly because of the modified immunoglobulins like HSI, thyroid stimulating immunoglobulin. So these thyroid stimulating immunoglobulins are acting like TSH. Thyroid stimulating hormone is the one which will bind with the thyroid gland, and it will stimulate the production of the thyroxine. But the thyroid stimulating immunoglobulin, which is a source which is produced from the immune cells, will also mimic like TSH. And it will bind to the same receptor on the thyroid gland to which the TSH binds, so that it will keep on stimulating the thyroid gland. As a result, the thyroid gland follicles becomes hyperactive. So this is called as a kind of hyperthyroidism condition. Both these are the autoimmune diseases, and these autoimmune diseases are subsided during the pregnancy in case of the female. If the female is already experiencing this kind of autoimmune diseases, during the pregnancy, these two diseases are suppressed. But once the mother delivers the baby, 3 to 6 months after the delivery of the baby, then those pre-existing autoimmune diseases can again be aggravated or exacerbated. This supports a view that elevated levels of sex steroids during pregnancy depress the immune response, mainly the progesterone. Okay, so here is experimental evidence in case of the mice. This has been done where the estrogen as well as the progesterone have been injected to mimic a situation of the pregnancy. in case of the normal females. So here is the wild type mice thymus gland, which is quite enlarged here, the control mice thymus gland. And here is the thymus gland of the mice, which was injected with estrogen as well as the progesterone. So here the concentration of these hormones was similar to those of the pregnant mice. So that there is involution of this thymus gland, which clearly tells us that this estrogen and progesterone are capable of diminishing the thymus gland activity. So the same situation can occur during the pregnancy also, where during the pregnancy number of cortical thymocytes are reduced, but not the number of medullary thymocytes. And here is the sectional view that you can see, where the cortex portion consisting of the immature T cells, whereas this is the medulla portion where the mature lymphocytes are found. But in response to the progesterone during the pregnancy or during the exogenous treatment in case of the normal female, so there is severe depletion of the T lymphocytes in the cortex region. So here is the experimental one where the mice treated with estrogen and progesterone, the cortex T lymphocytes are severely affected, whereas the medullary lymphocytes are not affected here, because the receptors for progesterone and estrogen are mainly present in the cells in the cortex region, so that they are highly responsive and they are distracted because of the high amount of estrogen as well as the progesterone. So there is a paper from this Dept et al. Entitled "Progesterone Receptors in the Thymus Are Required for Thymic Involution During Pregnancy and for Normal Fertility." Now let us look into the effects of endocrine ablations and the replacements on immune response. Endocrine ablation means the gland is being removed. The gland means in this case the gonads are being removed. It can be removal of the testes or removal of the ovary. And then the supplementation of the testosterone or estrogen can be provided to see what happens in the absence of the gonads, and when we replace the steroid hormones again, whether the effects are being restored with reference to the immune response or not has been studied. Okay, so here there is an experimental evidence which was done in case of the mouse, where the 2 months old mouse has got a very quite large thymus gland. And this is the sectional view, of course, the fluorescent stained one, the cortex portion as well as the medulla portion, where the 2 years old mice will exhibit the diminished thymus gland here. You can have a comparison here, the size of the 2 months old mice and the size of the 2 years old mice. So there is a drastic decrease following 2 years, and even the number of the cells are also being depleted here. And here to this 2 years old mice, the castration was done. The gonads were removed, and following 2 weeks of castration, there is enlargement in the thymus gland. And these lymphocytes in the cortex portion as well as the medulla portion are also being restored here. This is looking similar to that of the thymus gland, which was in 2 months old mice. So this clearly tells us that when this castration is done, the steroid hormones are eliminated. Means they are the source. The sex steroid hormones are being produced from the testes or the ovary. So when these hormones are cut off, then the thymus gland undergoes enlargement. As long as these steroid hormones are present, the thymus gland is suppressed. Once these steroid hormones are removed through the castration, then the thymus gland undergoes enlargement. In case of the males in other studies, like the thymus gland weight as well as the spleen weight also is shown to be increased following the castration. So there is a significant increase in the thymus weight as well as significant increase in the spleen can be seen here in this graph. And even these castrated male mice have higher number of CD4 plus, that is T helper cells, and CD8 plus. These are the T cytotoxic cells. So here this white bar represents the sham operated mice. Means it is a kind of operation where the operation is done, but no gland is removed, no organ is removed. These are called the sham controls. But just the animal is cut open and the stitches are done. The black bar represents the castrated one, where the animal is cut open and the organ is removed. That is, the testis is removed. So, following this castration, the immune cells number is significantly increased, particularly the CD4 plus as well as the CD8 plus is significantly increased as compared to the sham controls. Now, let us try to know about a condition which is intimately associated with the cancer in case of the males, where there's androgen and prostate. These two seem to be interconnected. From birth through puberty, prostate remains small and immature, while in post-pubertal males, prostate size increases up to 10 times than the prepubertal size. So this prostate gland is the accessory gland in case of the males, which is concerned with the production of semen. It also contributes to the semen. Most of the percentage comes from the seminal vesicles, but there are also other accessory glands which are also contributing to the quantity of the semen here, and that is a media which is very much important for the survival of the spermatozoa. So this prostate gland is smaller before the puberty, but during the puberty, the size is enlarged up to 10 times, because that also coincides with the highly active steroidogenesis in the testes. So this prostate gland has got androgen receptors through which the androgens can bind, and thereby they can control the activity of the prostate gland. So these androgens sometimes they may also trigger the carcinogenic development. So in such cases, the androgens can bind through the androgen receptors on the carcinogenic cells, and they can trigger the multiplication. So the oncogenic potential of the androgens is also being reported in case of the prostate cancer. So the prostate cancer can be treated by cutting the androgen supply. That is also another mode of treatment, where as long as the androgens are available, they can keep on binding with androgen receptors on the prostate tumor cells, so that they can keep triggering the carcinogenic development. So for this, there is a treatment method called as chemical castration. Because there are two methods here. One method is the testis has to be removed. So in such case, it becomes irreparable. Once the testis is removed, the androgens are also eliminated, so that prostate cancer can be controlled. Alternatively, retaining the testis, there is also another method called as chemical castration, where there is no need for the removal of the testis, but this involves the treatment of some hormones like LHRH agonists. LHRH agonists means luteinizing hormone releasing hormone agonist. These are also called as GnRH agonist, gonadotropin releasing hormone agonist. Agonist means it mimics the receptor for the LHRH or the GnRH. So it will bind with the same receptor to which the LHRH will also bind with the gonads. So when the patients receive these LHRH agonists for the first time, they may experience a phenomenon called as a testosterone flare. Because this LHRH is known to trigger LH in the pituitary gland, and LH in high concentration triggers the Leydig cells, because the Leydig cells have got the receptor for the LH, and then the steroidogenic potential is triggered, so that a high amount of testosterone can be produced. So this is called the testosterone flare. So this is in response to the LHRH agonist treatment. This happens only for the brief period. Following that period, the Leydig cells becomes unresponsive to the LHRH. Because of severe triggering, these cells are not able to produce any more testosterone afterwards. So then the possibility of subsiding of the prostate cancer can be expected. So there are also alternative treatment methods like the LHRH antagonists. So these are the one which are opposite to the LHRH, but they will also bind to the same receptor to which the LHRH will bind. but these will not exhibit any kind of testosterone flare, because these will bind and they will block the binding of the actual GnRH or LHRH, so that so there is no testosterone flare here, and the androgen production is also cut. So one such example is Jagarelix. So there are also some anti-androgens which can also be treated, like cyproterone, acetate, flutamide, and bicalutamide. So once the treatment is stopped, the androgen production usually resumes. So thereby these effects are reversible. So the chemical castration in case of humans are known to enhance thymus function, because following the chemical castration, there is a situation like the androgen is cut off. So as a result, there is significant increase in the total immune cells in the peripheral blood, and also there are significant increase in the total T cells, CD4 plus T cells, and also CD8 plus T cells. So these are all significant.
Madam, mute yourself.
Sorry. So these are all also significantly increased following the chemical castration. And even the natural killer cells are also significantly increased following the chemical castration because of the cutoff of the androgen supply. But the B cells are not affected here because they are not concerned with the androgen receptors. Now let us look into the hormonal regulation of the immune response, particularly the steroid hormones such as the estrogens are capable of influencing the target lymphocytes, because the lymphocytes are known to possess the estrogen receptors. They can bind with these lymphocytes and they can influence, and they can also bind with the thymic reticuloepithelial cells, where they can regulate the release of the hormones like thymosin, thymopoietin, so that they can influence the differentiation of thymocytes as well as the precursor cells. Estrogen treatment is known to suppress the cell-mediated immune response, but it is not suppressing the antibody-mediated immune response, because the antibody-mediated immune response is stimulated following the estrogen treatment, but the cell-mediated immunity is known to be suppressed following estrogen treatment. So this is a kind of differential response of the estrogen. So the estrogen treatment is known to inhibit the cutaneous delayed hypersensitivity reactions. So cutaneous delayed hypersensitivity means following exposure to some allergen, the body will respond, particularly the skin will respond to such kind of allergic substances, and this happens usually following 2 to 3 days. So that's why it is called a delayed hypersensitivity. Following 2 to 3 days time period, there is a severe response, immune response, which is characterized by the appearance of the rashes, as you can see here on the skin. So this is called as a cutaneous delayed hypersensitivity reaction. In such conditions, suppose if estrogen is treated, then it exerts the inhibitory effect on, particularly on the cell mediated immunity. Then the cancer immune surveillance. Surveillance means something monitoring, and these cancer cells are always subjected to some kind of mutation. So they, before turning into the carcinogenic cells, they convert themselves into the precancerous cells through the mutation. So normal cell turns into the precancer cell following a mutation, and in some cases these precancer cells do not exhibit the surface antigens which will make them to be detected by the immune cells. So in such cases, our immune system is not able to detect such kind of precancer cells. So this is called as immune escape. So thereby, these precancer cells transform themselves into the cancerous cells. Okay. So this kind of cancer immune surveillance is known to be inhibited by the estrogen treatment. So that will suppress the ability of the immune cells to monitor the recognition of the carcinogenic cells. And even these graft rejection responses are also known to be inhibited by the estrogen treatment. So in this picture, you will see that here there is a human skin allograft where on day 5 it is fully vascularized and the cells are dividing. But following those 12 days, there is a reaction. Our body is reacting to this allograft. So this is called as a graft rejection reaction, where these cells are being destroyed here. The implanted, transplanted cells are being destroyed. On the right side, suppose if the same allograft is transplanted for the second time. So now this immunological memory will be there in the immune cells. So these immune cells can rapidly destroy those allograft cells. That can happen within 7 days period of time. Earlier, it took 12 days to recognize that it is a foreign source. But second time, if the same allograft is transplanted, then the rejection can be quite rapid. So this can happen within 7 days. So this kind of graft rejection responses are also being inhibited by the estrogen treatment. So in women consuming oral contraceptives conjugated with estrogen or progesterone, the lymphocytes mitogenic activity is inhibited, because the oral contraceptives are mainly based on the disruption of menstrual cycle. So this is mainly because of the estrogen and progesterone that is a major ingredient within these pills, hormonal pills. So because of this, the high concentration of estrogen as well as the progesterone, they are known to be suppressing of the immune system. So the lymphocytes mitotic activity will be suppressed during the contraceptives intake period. Now coming to the androgens, both reticuloepithelial cells and certain classes of T lymphocytes possess androgen receptors, and they suppress the proliferation. Mainly, these androgens are also considered as the inhibitory to the immune response. The reticuloepithelial cells are found in the thymus gland, and T lymphocytes also possess the androgen receptors. So once this androgen binds with these receptors on the reticuloepithelial cells or the T lymphocytes, so then they will suppress the proliferation. And in situations where the chronic liver damage can occur due to hepatitis B virus, which is more in males than the females, where this androgen receptor is used by the virus to enter the T cells or liver cells. So in case of the males, the androgen receptors are present even on the liver cells, where the hepatitis B virus makes use of these androgen receptors as the entry gateway, so that it can act on those liver cells and it can cause damage to the liver cells. And the incidence of diseases associated with hepatitis B, like the Hodgkin disease, can also be associated with the hepatitis B. Hodgkin's disease is otherwise known as lymphoma. So this lymphoma is characterized by abnormal increase in the number of lymphocytes that you will see here in this picture. In case of the Hodgkin's disease, where there is a normal increase in the number of lymphocytes. And also the chronic renal disease can be associated with the hepatitis B, and the leprosy, etc., are also associated with the hepatitis B, which can be more common following this hepatitis B infection because of the high androgens. The virus may possess a surface antigen that cross-reacts with the androgen receptors in case of the males. And the progesterone is known as the immunosuppressive agent that we know through the pregnancy also, and during the menstrual cycle also. The immunosuppressive effect is mainly because of the progesterone. In studies involving progesterone-induced suppression of lymphocyte proliferation, the injected dose of progesterone levels was similar to that of the pregnancy. So the experimental condition where if you inject the progesterone, the dose which is similar to that of the pregnancy, you can expect the same kind of suppression of the immune response that may occur during the pregnancy. Now let us come to the glucocorticoids. Glucocorticoids are the hormones which are concerned with the stress response. So these are produced from the adrenal gland. like the corticosterone or cortisol. In case of humans, it is a cortisol, which is a major glucocorticoid. In case of the rodents, corticosterone, and even other vertebrate groups also, except for the fish and humans, others do produce corticosterone as a major glucocorticoid, and humans and fish are known to produce cortisol as a major glucocorticoid. So these are produced in the adrenal gland. Many investigators after causally have reported increased thymic weight due to adrenalectomy, because adrenal gland is a source of the glucocorticoid. Once this glucocorticoid supply is cut, then the thymus gland is known to exhibit enlargement. Suppose if there is a replacement of the glucocorticoids in the adrenalectomized individuals, then the thymus will undergo the thymus will again undergo hypertrophy as well as hyperplasia. This is in case of adrenalectomized animals, followed by replacement in the glucocorticoids. So this results in the thymic hypertrophy and hyperplasia. Hypertrophy means enlargement in the thymus gland. Hyperplasia means increased number of cells in the thymus gland. Ashwell and colleagues in 2000 suggested that glucocorticoids are essential at very low concentrations for early development and survival of the thymocytes. So we cannot altogether say that the glucocorticoids are detrimental to the immune system. Glucocorticoids are also required at low concentration for the development and survival of the thymocytes. Even for that matter, the corticosterone is produced in little quantities within the thymus gland. So this is other than adrenal gland, where the corticosterone is produced locally within the thymus gland, where it is known to play a role in the maturation of the lymphocytes. Thymic reticuloepithelial cells and lymphocytes do contain glucocorticoid receptors to which these glucocorticoid hormones can bind. And in recent studies, there is overexpression of glucocorticoid receptors in developing and mature T cells that can lead to decreased cell number in the thymus in adult mice. So these glucocorticoid receptors overexpression can lead to the decreased number of cells, thymic cells. Whereas a low concentration of glucocorticoids are required for the proliferation of the thymus cells. So this is again the concentrated dependent action that is observed with reference to the glucocorticoids. However, a decreased expression of glucocorticoid receptor is associated with the increased cell number in the thymus. So this is a kind of antagonistic pattern. Elevation of glucocorticoids due to chronic stress or therapeutic administration causes involution of the thymus gland. And again, here, if glucocorticoids are elevated, as may occur during the chronic stressful situation, or if there is a therapeutic administration where that may involve the treatment of the glucocorticoids, then that can lead to the involution of thymus gland. Enlargement of the thymus gland occurs in human patients with Addison's disease and in adrenalectomized rats. In adrenalectomized rats, already you know that glucocorticoid is cut off, so that can result in the enlargement in the thymus gland. In case of individuals with Addison's disease, this Addison is the name of the scientist, and this disease is characterized by hypocortisolism. Hypocortisolism means the cortisol level is lower in these individuals. So this can be because of the defect in the adrenal gland or the adrenal dysgenesis, which may happen during the embryonic development itself, where the adrenal gland itself is defective and is not able to produce sufficient glucocorticoids. So in such cases, it is called as Addison's disease. So this is a situation where the glucocorticoid levels are low, so that can result in the enlargement in the thymus gland. So there are also other hormones in the pituitary gland, like the somatotropin, which is otherwise known as the growth hormone, which is known to play a immunostimulatory role. So this because there are GH receptors, growth hormone receptors on these cells, so that the somatotropin can enhance both cytotoxic T cells as well as helper T cells in normal thymocytes in cortex region of the thymus. The cortex region lymphocytes are immature and contain GH receptors compared to the mature lymphocytes in the medulla. And here is a situation where the runt disease which is also known as graft versus host reaction, where if any transplantation is done from a recipient's tissue into the donor, for example, in this case, the bone marrow transplantation. The bone marrow or the peripheral blood stem cells transplantation is, if it is done to the donor, then the recipient's bone marrow will recognize the donor's body as the foreign one. So the recipient's body is under the attack by the grafted tissue. So this is opposite to what happens when the graft is being rejected from our body. So there, our body is recognizing the allograft and it is rejecting it. But in this case, the grafted tissue itself is acting on the host tissue. That is in the donor's body itself. The grafted tissue here is the transplanted bone marrow. So that will act on the recipient's body itself, thinking that it is the foreign body. So then there can be severe reaction. So that's one such picture is shown here, where the person is exhibiting this graft versus host reaction, also known as the grunt disease, following the transplantation of the bone marrow from the other person. So under such circumstances, if GH treatment is done, that exacerbates this disorder. That means it is immunostimulating in function. If GH is administered to these kind of individuals, then this reaction will be further aggravated. Okay, so the hormone-like activity of the thymus derived factors has been identified since from 20 years. It's with reference to the hormones of the thymus gland. There are 4 hormones that have been identified in the thymus gland. Thymopoietin, which is of 49 amino acids, thymolin, which is of 9 amino acids, thymosin alpha 1 of 28 amino acids, thymosin beta 4 of 43 amino acids. All these hormones are known to be involved in the differentiation of different types of T cells. So the thymus gland acts like the endocrine gland because of the production of these hormones, which are capable of exerting the endocrine function. And it also acts as an immune organ. So there appears to be some connection with other organs also. So we do consider this as the axis, like a hypothalamo pituitary gonadal thymic axis, which establishes the connection between hypothalamo pituitary gonadal axis with the thymus. HPG axis classically is known as the axis which controls the reproduction. And now that is connected with the thymic axis here, or the thymus gland. So the thymic hormones such as thymosin beta 4 can cause increase in GnRH amount, which can increase the sex steroid secretion by gonads through LH and FSH, which forms a self-regulating cycle. So these thymic hormones are known to perform a stimulatory action on the GnRH, by which they can also increase sex steroid hormone, and increase the sex steroid hormones can negatively affect the thymus activity. So we can see how this is being regulated here in this chart. Okay, so here the hypothalamus acts on the GnRH, and the GnRH cells will produce, the GnRH hormone will act on the pituitary gland, where this FSH and LH are being produced from the pituitary gland. And this LH mainly acts on the gonads to stimulate the steroidogenesis, that is testosterone in case of the males, and estrogen in case of the females. Suppose if the sex steroid hormone levels are lower, then this will stimulate the thymus gland. Low sex steroid hormone levels are known to be stimulatory on the thymus gland. So the thymus produces thymosin beta 4, and this thymus beta 4 is having stimulatory effect on the GnRH. So that now this GnRH is again increased, and again it stimulates the gonads through the LH, so that the sex steroid hormone can be increased. And also the lower sex steroid hormones can also give the negative feedback to the hypothalamus, where it can directly act on the GnRH. This is another loop through the HPG axis always will be operating. And this particular loop is through the thymus gland. So the thymus gland, in response to low sex steroid levels, can stimulate the GnRH through the thymosin beta 4. And on the other hand, they can also stimulate the lymphocyte function through the thymosins. where the lymphocyte proliferation can be increased in response to low sex steroid levels. So here is another situation where the sex steroid hormones can be increased. So suppose if the sex steroid hormones are increased, then also through the negative feedback mechanism, they can act on the GnRH as well as the pituitary through the negative feedback mechanism. This is the mechanism of the HPG axis. But here the mechanism concerned with the thymus gland, the increased sex steroid hormones will inhibit the thymus gland activity, where the release of thymosin beta 4 is decreased, so that the GnRH stimulation is decreased here. And the thymus gland also produces a low amount of thymosin, so that the T cells proliferation is decreased. And in addition to this mechanism, estrogens or testosterone can also directly act on these T cells, because the lymphoblasts are known to contain, lymphoblasts means immature T cells. So these lymphoblasts are known to contain estrogen receptors as well as the testosterone receptor, I mean the androgen receptors. So they inhibit the maturation of the T cells by directly interfering. So this is a kind of a circuit, self-regulatory circuit, which is occurring between the HPG axis as well as the thymus gland in response to increased sex steroid hormone levels or decreased sex steroid hormone levels. Now the interaction between the HPGT and HPAT axis. HPAT stands for adreno hypothalamo pituitary adrenal thymic axis. HPGT, hypothalamo pituitary gonadal thymic axis, that we studied about that. And this is another axis that involves the adrenal gland here, and you know that adrenal gland produces glucocorticoids. And during the stress, adrenal gland is known to produce a high amount of glucocorticoids. And these high amount of glucocorticoids are known as immunosuppressive in nature. And that lowers the sex steroid hormone levels. And these lower sex steroid hormone levels are known as the immunostimulatory. So there's a kind of loop that is operating here. Increased glucocorticoids following stress are immunosuppressive, and that will suppress the sex steroid hormone level also, because it is experimentally proven that the increased glucocorticoid concentration suppresses the sex steroid hormone level. So the sex steroid hormone level, decreased sex steroid hormone levels are now immunostimulatory. So on one hand, there is an immunosuppressive effect of glucocorticoids. On the other hand, there is an immunostimulatory effect of sex steroid hormone levels. Now these two axes are a kind of balanced one. So the kind of balance or homeostasis is maintained during the stress. So it acts as a self-regulatory mechanism on both developing and mature lymphocytes to prevent excessive depression of immune response. So here,
Dr. Ganesh, can you take up some questions? Dr. Ganesh, can you take up some questions?
Yeah. This is the last slide.
Okay, sir. Okay.
Thank you.
Okay. Participants.